My name is Mohamed Sobeeh. I am a PhD student in Cairo University, Faculty of Physical Therapy, and today I will present a topic related to altered brain structure in people with carpal tunnel syndrome, systematic review and meta-analysis. First, this study registered in Prospero database for complete protocol. You need to read the complete protocol before implementing this study. Carpal tunnel syndrome is a common entrapment neuropathy occurred in the upper limb because of focal pressure in the median nerve passing in the hand. The main symptoms are pain and or paresthesia. Prevalence of carpal tunnel syndrome in females is larger than in males. So the prevalence in females are 3.8% and in males 2.7%. The interesting thing that uh, uh, despite the carpal tunnel syndrome is a peripheral neuropathy, there are many reports showed evidence of central changes in carpal tunnel syndrome. Like there are extramedian spread of symptoms detected in people with carpal tunnel syndrome. Median nerve usually supply lateral two-thirds of the hand and lateral three and half fingers. Many reports showed extramedian spread of symptoms. I mean that the symptoms should be restricted to the area supplied by median nerve. But in many people with carpal tunnel syndrome, we showed like extramedian spread, like in the shoulder, in the arm. There are sometimes people you can find some people complaining from pain and paresthesia in the arm and shoulder. Also, uh, some reports showed limited or disrupted descending pain modulation, what's called conditioned pain modulation, disrupted in people with carpal tunnel syndrome. Other reports showed presence of thermal and pressure hyperalgesia. Thermal and pressure hyperalgesia post topics are signs of what's called central sensitization. I mean, central changes occurred in spinal cord and brain after this common peripheral neuropathy. These signs indicated there are some changes occurred in spinal cord circuit and brain gray and white matter. Evidence of central changes also, there are some studies showed changes in MRI and fMRI regarding the gray matter. There are reducing gray matter of somatosensory, pulvinar, and frontal areas. Also, the intercortical, interdigit cortical separation distance of index and middle finger showed some sort of reduction. Unfortunately, people with central changes, with people with carpal tunnel syndrome and central changes, showed limited response to conservative management and usually refer to surgery. So, the purpose of this study is to confirm the all brain structure and function based on previous studies using imaging modalities, MRI and fMRI, to answer our question, which is, are there alterations in brain structure and function in people with carpal tunnel syndrome to confirm the central changes happened in these people, on these people? The method, we, we searched like 5 databases in Embase, Web of Science, Scopus, PubMed, and Cochrane, with eligibility criteria of studies should involve adult people with carpal tunnel syndrome. The stages of the disease in these people are subacute or chronic. Also, people should be diagnosed through clinical and or electrophysiological assessment using nerve conduction studies. The third eligibility criteria is those people with carpal tunnel syndrome and the healthy control group should be investigated using MRI or fMRI to investigate two main variables: interdigit cortical separation distance and cluster size related to digits in somatosensory cortex. This is our flow diagram. The main important part is the included studies in qualitative assessment were 9, wherein quantitative and meta-analysis were 4 studies. We implemented risk of bias assessment using Newcastle Ottawa quality assessment, which is directed toward observational case control studies. We searched only for observational case control studies to compare between carpal tunnel syndrome and healthy control. And Newcastle Ottawa quality assessment used mainly for assessment of case control studies. We can show that our four selected articles in quantitative meta-analysis are of high moderate quality. يعني 5 from 9 are good quality, not bad quality. But only one studies represent a bad quality, which is for Haddouche et al. 2017. To illustrate my results, I quoted a picture from Nabado et al. 2006, illustrating center of mass of second digit and third digit, both second and third digit supplied by median nerve, as well as fifth digit, center of mass of fifth digit in somatosensory cortex. Fifth digit supplied by another nerve called ulnar nerve. Interestingly, our results showed that the distance between the second and third finger supplied by median nerve were significantly reduced in carpal tunnel syndrome people compared with healthy control group. Also, the distance between the second and fifth digits were significantly reduced in people with carpal tunnel syndrome in comparison with healthy control group. The final result were the interdigit cortical separation between the third and fifth digit were not significantly reduced. So here are my results. Interdigit cortical separation distance, we used what's called the standardized mean difference. That was 0.8. 0.8 is a big effect size here. The effect size here is not small, large effect size, indicating significant reduction regarding the interdigit cortical separation between the index and middle finger. Also, the interdigit cortical separation of index and little, second and fifth fingers also significantly reduced with the standardized mean difference of 0.84, also large effect size. While the interdigit cortical separation between the third and fifth finger showed no significant difference. Our second variable is the cluster size. Cluster size, there are two high quality studies showed enlarged activation of somatosensory areas responsible for index and middle finger. It means while after inducing some sort of tactile stimulation in the index and middle finger, there are blurring excessive activation of somatosensory areas responsible for the second and third fingers. This can explain the reduction of the distance between those two fingers. Also can explain the reduction between the second and fifth fingers, interdigit cortical separation. So here are a picture that can show the activated or blurring or enlarged cluster size of areas responsible for index and middle finger. As we can see in the left picture, this picture related to healthy control group, and the right one related to carpal tunnel syndrome group. Simply, the red line represents the cluster size of index finger, while the green one responsible for or represents the cluster size of middle finger, while the blue line represents the cluster size of little finger. I think this picture shows the enlarged cluster size of index and middle finger in carpal tunnel syndrome compared with healthy control. Our conclusion: altered cortical morphology and function, as evident by increased cluster size and decreased interdigit separation of index and middle finger, proves the central mechanism involved in carpal tunnel syndrome. So, what's the implementation of this study? As I said in the first part, people with central changes usually showed limited response to conservative treatment and usually referred to surgery. This can increase the cost of treatment for those people and health insurance also. So, if we search it for new interventions that can target the somatosensory changes occurred in such peripheral neuropathy, we can improve the outcome and the quality of life for in those people. Also, we can try some sort of treatment like pain neuroscience education, cognitive behavioral therapy, like transcranial electrical, uh, transmagnetic stimulation of somatosensory cortex, like direct current stimulation, somatosensory cortex. All of these methods can be used in the future, trying to improve the outcomes in people with carpal tunnel syndrome. So reducing the cost, health insurance cost, improving quality of life, improving the functionality for those people. This work cannot be done without the effort of my research team. So thanks so much. And I am ready for any question.
So thanks a lot, Mohamed, for the interesting talk. Do we have any questions for Mohamed? So I have a quick question. So in your slide 13, you mentioned altered cortical morphology. So what exactly do you mean? Because with fMRI, you can't look at morphologies of neurons in the cortex. You probably mean cortical geometry.
No, some studies use diffusion tensor imaging to
Okay.
study gray matter and white matter, and they
Okay.
showed like reduction in the gray matter, but I couldn't this data in meta-analysis. So I will refer it in some sort of qualitative assessment because of I can find only I don't remember actually two studies or one study represented reduction in gray matter and white matter using diffusion tensor imaging, but I couldn't add them in meta-analysis.
Okay, sure. Yeah, no problem. Yeah,
Thank
that's
you
good
so
to
much.
know. So